Hello and good morning to those that are online. This is Mark Congreve from ICAN, and welcome to this morning's GRDC sponsored webinar. And today we're going to be talking about the ecology and management of wild oats. This webinar series builds on some of the ones that we've previously conducted. For those that haven't been following our webinar series to date, we've already run similar webinars on other northern key focused weeds such as feathertop rhodes grass, south thistle, barnyard grass, etc., wild radish, fleabane, and you can find previous recordings of those webinars on either the GRDC YouTube site or you can get links to those through the ICAN site where you would have registered for today's webinar. For each of these weed focused workshops, we've work through a bit of an understanding of the ecology of the weed, then move into where we're at with our latest understanding of resistance, and then we move on to the management tactics that we need to implement to keep those weeds under control. So a good resource that we're building up over time for each of those key weeds. So what I want to do now is just move through the presentation. As we talked about, I'll start with the biology and ecology side of things, talk a little bit about resistance updates, get on to management tactics. So if I start with the biology and ecology and the key things that we need to understand about wild oats, first one is we're dealing with two different species. And you can see there from the graphs, they occur slightly differently across Australia, but in a lot of situations, they're going to be in the same paddock at the same time. In early season, really difficult to distinguish between the two different species. So Avena fatua, more dominated if we're in South Australia or Western Australia, it's most likely to be the species we're dealing with. If we're in the southern eastern states, we could be dealing with fatua or ludo wild oats. If we're getting into Queensland, it's more likely we're going to be confronted with ludo. Key difference, as I said, very difficult to physically tell apart in the early growth stages, but there is some evidence in the research that Avena fatua may tend to germinate and therefore drop its seed a bit earlier as well. So that has implications as to controlling late season emergences, and I'll talk a little bit about that as we work through this presentation. Looking at the seed production of wild oats, so fairly large seed produced. I think most people are familiar with what wild oats looks like. The research would show that we can produce up to over 200 per plant, and if you look at some of the high populations that can occur with wild oats, that can result in potentially 20,000 wheat seeds per metre square being produced under dense infestations. Obviously, we don't want to let it get to that stage, but it can produce a lot of seed in a single season. As far as the germination patterns go, usually we see a large flush in late autumn, early winter, and the research would show that maybe approaching 40% of the seed bank that's in the soil. You then tend to see some later germinations. Those later germinations generally are often not as competitive, but they're typically the germinations that may replenish the seed bank the next year, particularly if they're not controlled. So we really need to focus on controlling this species at the start of winter, but also through the crop. And again, I'll talk about that a little bit later in the presentation. Being a relatively large seed, it will germinate from various different depths. So it will germinate if it's left on the soil surface. It loves germinating from shallow depths around 3, 5, 7 centimetres, but potentially can also germinate from deeper than that as well. So that has implications if we're putting cultivation into the paddock. As far as seed bank persistence goes, we do get a lot of the seed germinating the year after it was shed. So, the information there would suggest that maybe 75% can germinate in that situation the year after it's shed, more germinating the second year after it's shed, and if we get no seed bank replenishment at all, for 2 to 3 years, we can generally drive that seed bank down very low. So we really need to be focusing on those escapes. It's those few escapes that we see in the paddock sticking their heads up above the crop. They're the ones that are really building the seed bank ongoing for the next year or two. So we really need to focus on getting those out if we want to drive that seed bank right down close to zero. I mentioned cultivation before. I guess my thoughts around here is we've got two different systems. So in older full cut cultivation systems, what we're doing is we're really mixing the seed throughout the soil profile. So when we developed control strategies with products like trifluralin or triolate, when they're used in conjunction with full irrigation and aggressive cultivation and incorporation into the soil, they work really well because they're getting the herbicide down to where the seed is in the soil. On the downside of using full incorporation systems, we're going to increase the seed persistence once it's buried, in line with most species, that once we bury the seed actually retains its viability for a longer period of time. In more recent years, we've moved towards zero-till farming systems. If we're going to operate in this system, and particularly if we're in a transition from cultivation to zero-till, just be aware that it's probably going to take 3 years at least to get rid of those seeds at depth, so they're not viable anymore. So there's about a 3-year conversion to getting all those seeds towards the soil surface in a zero-till system. But going hand in hand with those zero-till systems, there's been some newer claims developed over recent years to match some herbicide use patterns with that farming system. So in the last 12 months, 18 months or so, there's been a change to the Avadex label to introduce higher application rates applied under a incorporate by sowing incorporation strategy for a range of different crops on the Avadex label. So that's a relatively new claim to align with these IBS zero-till farming systems. Trifluralin, been around for a few years now at higher rates, again in that use pattern, and again in the last couple of years there's also been a registration for Sakura. And I guess one of the key things is that these zero-till use patterns are probably most likely to give you high levels of suppression, but probably unlikely to give you complete control, mainly because of where the weeds are positioned in relative to the herbicide. And I'll talk about that a little bit later on. So looking at the next thing I want to touch on is the importance of wild oats to competition and how it impacts on particularly yield, and in this case yield of wheat. So you can see here, again, this is an old data set, but it still holds very true, that under relatively high weed pressure, so blue line being 50 wild oats per square metre, red 150, green 300 wild oats per square metre, so very high populations. If the wild oats gets out, say 6 days before the wheat, gets out of the ground 6 days before the wheat, we can see that we're experiencing 50 to 85% reduction in yield. So extremely competitive in that situation, particularly under high pressure. It's emerging the same time as the wheat. There's a little bit more competition coming on from the wheat there, but still getting 30 to 65% reduction in potential yield. And if the cereals get a 6-day jump on the wild oats coming out of the ground, there's obviously a reduction there in the loss, but we're still getting from 15% yield penalty in that situation. So a highly competitive species when it emerges early with the crop. So that's some old data. Some more recent data conducted by Bugarath last year up in Toowoomba in 2015. So this is a glasshouse trial, but there's a lot of data on this slide, but I just want to walk through the key points because I think it's really interesting. So in this situation, we're talking about a relatively low density. So this was a pot trial, but it equates to about 8 wild oats per square metre, so a lot lower densities than the previous slide. But what we can see here is the wild oats were planted at different timings to emerge at different time frames after the wheat had emerged. So at our first data point, the wild oats are emerging 4 days after the wheat's already out of the ground, and then you can see it continues 7 days, 9 days, up to 39 days after the wheat has already emerged. So if I look at the height of wild oats, this darker blue line, there's really no significant reduction in the height of wild oats up until about the 17 days post the wheat emergence, and then the wheat's become really competitive. You can see that there's a big fast decline in the height of the wild oats, and by 39 days wild oats couldn't actually establish due to competition from the wheat. If I look at the red line, the biomass of the wild oats, while the height hasn't necessarily been affected early days, the total biomass has been. So there's competition from the wheat emerging straight away, and you can see it's declined fairly linearly as we've increased that time delay after the wheat's emerged. Likewise, the tiller numbers, the green lines, that's reduced in accordance with the increase in competition. The head number was also recorded, so that's your purple line through there. And if you look at the lighter blue line, this is actually the seed number. This is one I'd really focus on. You can see that as competition from the wheat has increased at each time point, the seed number produced has dropped away really fast. And by this point at 27 days, there's not much seed being produced at all, and once we're getting out to 32, 39 days, the crop competition has almost eliminated, or in 39 days has eliminated, the seed production. So I guess the key take-home message there is wild oats can be very competitive early. Wild oats will be competitive early, your crop can be competitive later. If we look at what happened to the wheat in that same trial, so this blue line through here again is the wild oat biomass, just for reference side of things. But looking at the wheat, the other lines, the wheat biomass increased over time as it became more competitive with the wild oats. Likewise, the wheat tiller numbers, the red line, the head numbers, the green line, also slightly increased over time as well. And if we actually look at the calculated yield from there, with no competition from the wild oats, it equated to an average of 5.5 tonne per hectare, about a 20% yield decline in this trial where the competition commenced from the wild oats at 4 days after application. So again, backing up that earlier research that these wild oats can be very competitive early season. Moving on to resistance, I just wanted to touch on a few different things. So quite often we've seen the level of ryegrass emergence in the same paddock when it's treated off with the same herbicides emerge a lot faster, or present a lot faster than what it does for wild oats. Why is that the case? So this is a really interesting study from ARI over in Western Australia, which was published in Australian Grain a couple of months ago, just looking at the differences in the two populations. So if we look at ryegrass, it's an outcrosser, so it cross-pollinates with other ryegrass out in the field, and it has the pretty standard diploid genomic structure where there's 2 copies of each gene. If we get a mutation on a gene causing resistance, that's got a 50% chance of being outcrossed in each outcrossing event. So relatively high chance and high frequency of expression of that gene, and moving it on through the population. If we look at wild oats, it's hexaploid. So for each gene, there's 6 different copies. So if we get a mutation on 1 copy of the gene, there's another 5 copies out there that don't have that mutation, at least initially. And so therefore, generally the resistance is expressed at a lower level. And as it's self-pollinating, means that it's going to take a lot longer for that resistance to spread across the paddock. So there is differences between the 2 different species, which explain why resistance can take longer to occur with wild oats. We have to try a bit harder to create it. If I look at the resistance status, I won't spend much time on this graph, but this is just the historical data extracted from the Charles Sturt University testing over many years and extracted from their reports. Our FOP herbicides have had high levels of resistance for basically as long as we've been tested, running up in the 70 to 80% levels. You can see the lighter blue lines, the Maximising Group Z. It's bouncing around a bit, but we're starting to get quite significant levels of resistance occurring there. The green lines being panoxidine or axial. Again, we've got concerning levels of resistance present in a number of populations. And the other two lines on here, the Group B herbicides and the DIM herbicides, some level of resistance there, but seems to be at sort of relatively low levels and seems to be sort of consistent over the years, where it's just bouncing around at those low levels. Come back and talk about those in a little bit more detail. If I just look at the 2015 data, so again, FOPs, more resistant populations than not. DIMS, only one observation last year from 56 samples that were in that test. Talked about the DENs, there's some level of resistance there, the Group Bs, the Maximising, the other ones that weren't on that previous chart, Avadex, glyphosate, atrazine. While some of those testing numbers are fairly low, no resistance detected to date. So the key thing is really to understand for our individual population, what are we actually dealing with as far as resistance goes. So testing really important. So if I put that together and start moving on to how we're going to manage this, it basically means we're going to have to require multiple tactics stacked together. So because of this, we've got a highly competitive crop, particularly early season, and we've still got these late season germinations coming through, multiple tactics stacked together. So one way to do it is an early season control, either a pre-emergent or possibly an early emergent. You stack that with crop competition that we've already talked about, and then some form of late season control, we can drive that seed bank down. The other potential way we can manage wild oats is to take the crop out of winter crop production, going with a non-selective strategy over winter, relying on glyphosate or paraquat, which to date we don't have any resistance, and potentially moving to a summer crop for a couple of years. And if we do that, we can drive down the seed bank relatively fast. So I'll talk about those in a little bit more detail. Crop selection, already touched on this, just want to reflect on it again. You can see here in this trial, this old data but will still hold true, that wheat, having our populations get up around this 100 plants per metre, the actual wild oat seed production can decrease substantially. In this trial, barley was much more competitive than wheat with that early season vigour. So again, choice of crop can be important. Something like chickpeas, which are going to be on wider rows and more thin early season, are going to be even a poorer competitor for wild oats. And we also have a lack of in-crop herbicide options for chickpeas in many situations, particularly where Group A resistance presents itself, and that can be a big challenge and leading to escapes, which we often see in a number of situations. So crop selection can be very important. If we talk about pre-emergents, we've got a range of different pre-emergents available to us across a range of different mode of actions. When we're dealing with pre-emergents, we need to keep in mind we're dealing with a large seed. It may be coming from depth, and we've got these staggered germinations throughout the season. So it's unlikely we're going to get 100% control with a pre-emergent, so probably going to need another tactic stacked on top of it. Haven't got time to talk about stubble binding with some of these herbicides that tie up on stubble, or how we need to manage those, but there's a lot to understand to make those herbicides work properly. I've just listed here, and we can come back to this recording later, I've just listed the different registrations that are available to us as far as pre-emergent herbicides by mode of action. So we've got a range of different Group C herbicides, Group D, Group J and Group K as we move down there. The ones that are in blue are the relatively new registrations in the last year to two. So there's a new IBS registration for use of turbine pre-cereals. Rustler in canola is a relatively new IBS registration as well. High rates of Avadex on the new label for IBS application is another relatively new claim, and Sakura has been out for a couple of years now for suppression in the crops that we can use Sakura in. So again, some new changes to registrations which assist us in the field. I just wanted to quickly touch on this slide. This just shows what happens, the difference between wild oats and wheat, and explains a little bit about how pre-emergent herbicides, particularly products like trifluoralin, work in that scenario and why we don't get 100% control in all situations. So if we've got our herbicide incorporated into the soil and we've got a soil treated layer and we've got our seeds down at some depth, what happens in a crop like wild oats is at emergence this mesocotyl expands, it pushes the coleoptile node up, and the coleoptile node is probably the main part where trifluoralin enters the emerging seedling. So that coleoptile node is pushed up into the herbicide treated area and enters the seedling in that way. For a crop like wheat or barley, the coleoptile node, the mesocotyl doesn't expand, and the coleoptile node remains very close to the actual seed down at depth. And it's this spatial separation as providing this coleoptile node is well below the treated band of herbicide. That's where we get our selectivity, and we're able to use products like trifluoralin in wheat and barley. The point I want to make there is, when we've got our wild oats just sitting on the soil surface and we're trying to get the herbicide in through the coleoptile node, we either need direct application of the herbicide onto the seed, or we need to rely on root uptake, which doesn't work very well for products like trifluoralin. So hence that's why we tend to get only suppression from those surface germinating weeds. Just touch on Group A herbicides. Probably everybody's relatively familiar with Group A herbicides. We've got a range of different Group A herbicides that we can use either in broadleaf crops or cereals, and different properties depending on the different subgroups that are there. I'll come back and talk about that from a resistance point of view in another slide, but looking at it from I thought this slide was interesting and worth putting in. So again, looking at the crop competition between wheat and barley, we're seeing without any herbicide the same trends that we saw earlier in some of those graphs, that barley is going to be more competitive than wheat. But in this trial, what was also stacked on top of the crop competition was different rates of herbicide achieved was used in this situation, and this would have been when achieved was still no resistance occurring. And you can see that stacking the herbicide at full rates on top of the different crops gave us extremely good control. But even at lower rates, we still had some additional benefit from stacking the crop competition and the herbicide together. Resistance, just touching on the resistance to the Group A herbicides again from that Charles Sturt data. So if you're looking at the FOPs, we saw that data earlier, is that blue bars here, many resistant populations out there, but there still are some populations which are susceptible. If we're looking at the DIMs, we've got your mostly susceptible populations out there, and axial a mix in between. So we need to resistance test to know what we're dealing with. I just wanted to touch on this data as well. This is a fairly complex chart that's there, but I'll just work through this in a number of different situations. So this is some work out of NGA conducted in 2014 in chickpea trials. So we had 4 different trials here with different levels of resistance across the populations. So if I just compare the trials, which are the purple circles, and the Bowenville trial, the green triangle. So in this situation, we've had our verdict of our 2 application rates working quite well. So we've got a FOP susceptible, 2 populations here that are obviously FOP susceptible. If we look at clethodim or status in this trial, again across those 2 trials, lower rate of 250 mils, not quite as good, but at 500 mils, very good control. And that's reflective of what we've probably seen over the years, that generally in a susceptible population, the FOP herbicides traditionally work slightly better than the DIMs, or a bit more robust than the DIMs, and hence that's why they've been the product of choice early on. If we compare our 2 populations, where now verdict has given us poor to very average levels of control, so obviously we're seeing Group A resistance or FOP resistance emerge in these 2 trials. You can see there that the verdict has basically failed. The DIM, in this case clethodim, has given us slightly better control at the low rate, but still relatively useful control, but not 100% control at the higher rate. So as we're starting to see Group A resistance emerge, we're still seeing slightly more useful control from the DIMs in that particular trial scenario. What was also interesting here is if you look at across the 4 trials, the susceptibles and the resistance, the verdict at 100 mils, status at 250 mils, neither of those were consistently robust across the 4 different populations. But when they were tank mixed together, that gave us an increase in control and probably getting up to something which may be acceptable in the field. The other thing that was really interesting from this trial was that the low rate of status was mixed with liase. Now, this wasn't in a hard water environment, this was in a rainwater environment, and there was a substantial increase or reduction in variability of the control received with liase, even in the absence of hard water. Most likely the ammonium ions in liase are helping to get some sort of additional penetration through the leaf. So that's an interesting observation if you're using clethodim against wild oats. So I've probably covered off on all this. I've mentioned a few times resistance testing to make sure we know what we're dealing with, really important. If we're going to use our Group A herbicides, we really need to maximise the coverage and treat small weeds, keep our water rates high, largely treat these as more like a contact type herbicide because they don't translocate particularly well. Make sure we're using the correct adjuvant as recommended by the manufacturer. Would always recommend, as we're moving into low to moderate levels of resistance, that we're keeping our maximum label rates that we can use, keep right up to the top of the label rates. Avoid broadleaf tank mixes. They can reduce the performance in some situations, and I wouldn't rely on these as the only tactic. They're probably good, useful tactic to be stacked on to some of the other tactics that I've been talking about. We've also got options of Group B herbicides in our cereal crops. So I've just listed those that are registered there. Again, haven't got a lot of time to go into these in detail, but they are not always 100% selective to the crop, so we can potentially have some crop issues. So we need to consider, make sure we're timing them at the correct growth stage, make sure we follow the adjuvant advice on the label, and watch for some of the tank mix partners which can hold up some of those herbicides, but if they're used properly according to the label, they should be fine in most situations. And for control of wild oats, we really need to target small weeds, make sure our conditions are really good, and these herbicides tend to work best when our densities of wild oats are low and struggle a little bit when we're just swamped by really high populations. But if we do all that, they can work really well. And, these Group A and Group B herbicides probably fit best on system following a pre-emergent herbicide to clean up any escapes and some of those later season germinations, probably where we should be thinking about targeting those. Resistance status, very small numbers to Innovix, Crusader and Hussar in last year's testing. Some level of resistance there in the Atlantis populations. Again, comes back to knowing what we're dealing with, which comes back to getting a resistance test done. If we don't like any of those options, we do have the option of herbicide-tolerant crops. So we've got clearfield, canola, barley, wheat available to us. That gives us the option of some of the Group B IMI options, such as Innovix or On Duty. That can work in situations where we're dealing with Group A herbicide resistance, and some growers are going down that path. We also have the option of triazine-tolerant canola, particularly to get Group Cs into the mix if that's the way we want to go, to try to get some rotation of resistance around. Generally, they're probably only going to give you suppression in that situation. The new turbine label does have a claim for early post-emergent control in those in TT canola, so that might be something that's of use as well. Getting towards the end of what I wanted to cover off on today, spray topping. We have the option here for those that aren't familiar with spray topping of coming along with a later herbicide application to largely try to sterilise the seeds. The key point is to get the timing right here. So we've got two different mode of actions we can use for us. Flamprop or the old Madivan product, and the various different versions that are out there now, being a Group Z. You look at the key timing according to the label is applying in cereals up to the start of booting or growth stage 40, and ideally targeting the wild oats at 30 to 40 growth stage. So these aren't going to really give effective control when we're going out and targeting large plants that are fully out in seed head. We've got to get onto them still relatively early. The other options are axial, anoxidin, label slightly larger from the cereals out to the end of booting rather than start of booting, and the wild oats, according to the label, we can target slightly later, but still not a particularly late application. We really need to get those herbicides on because once we're starting to get out into these really late growth stages, out in growth stage 61 and above, the performance of those herbicides starts to drop off relatively fast in that situation. So follow the label advice and keep the applications where they need to be. And I think this is the last slide. Just wanted to touch on some of this work from last year out of the Glasshouse trial that Bugarath conducted which earlier we used in the Ecology situation. What he did here was also looked at the height of seed production of the wild oats, thinking about can we actually use harvest weed seed control for wild oats in this situation. So we're talking Ludo wild oats in this particular trial, and you can see that as the time to emergence got increased, the plants got shorter and shorter and shorter from the competition. And we measured here the amount of seed produced above 15 centimetres, which is potentially seed which could go through the front of the header, versus that seed that's produced below 15 centimetres. So in these earlier timings, there's still a significant amount of seed produced which could end up in the header and potentially available for some sort of windrow burning or harvest weed seed destruction of some sort. So, we do have the thought process that wild oat sheds its seed, but, in some situations it still may be a candidate for weed seed destruction. A bit more research required in that space. So I've probably just about come in on time for my 30 minutes that I wanted to present for today. I know I've pushed through that fairly fast. There's probably a number of questions out there. Certainly like to thank NGA in particular and Coffee that have provided their data for this, and a range of other data sources that I've acknowledged throughout this presentation, which has hopefully given some indication of the available research on wild oat control and management and ecology, and thank GRDC for allowing us to put on these webinars as part of our integrated weed management extension project that we're running in the northern region.